Hello everyone, I'm Sangeeta Dwivedi, Assistant Professor of Modern Institute of Pharmaceutical Sciences, Indore. Today's topic is Microscopy Introduction part. So microscopy is the technical field of using microscope to view objects and area of objects that cannot be seen with the naked eye. In other terms, we can say that microscopy is defined as the use of a microscope to magnify and study the small objects that are too small to be visualized with the naked eye. There are two types of microscopy is there. First is optical or light microscopy and other is electron microscopy. So with the help of our naked eye, we can say the smallest thing which can be a size of 0.1 millimeter. With the help of light microscope, we are able to see a 0.1 micrometer things which can be a size of 0.1 micrometer, and with the help of electron microscope we are able to see those things also, or those areas also, which have a size of 2.5 nanometer in their sizes. So clearly visible that with the help of electron microscopy we can able to see the smallest area and portion and things of various cells and uh, various cells. So in light microscopy, light passes through a specimen through a single or a series of magnifying lenses to allow a magnifying view of the sample. In case of electron microscopy, electron beam passes through a specimen and give a very magnified and resolved view of sample. There are three basic factors which are involved in microscopy and are very important when we are talking about microscopy. That is, first is magnification, then second is resolution, and lastly is contrast. So let's talk about magnification. Magnification is a measurement of how much larger a microscope uh, causes an object to appear. For instance, the light microscopes typically used in our college's lab magnify up to about 400 times of actual size of any object. So total magnification is achieved by the magnification power of lenses which are using in that particular microscope. That if we are saying that magnification power of a microscope, then it is equal to the magnification power of a objective lens and magnification power of ocular lens. So for an example, if we are saying that the magnification power of objective lens is 40 and the magnification power of ocular lens is 10, then the total magnification power of a microscope is to be equal to 400. In simple microscope, the magnification power is about 1000 to 1500, whereas in the case of electron microscope, it can be greater than 2,50,000. So clearly, differentiation of magnification power are mentioned here between simple compound microscope. Now the next is resolution. So resolution is the ability to see two close objects as two distinct objects is known as resolution. The limit of resolution is the closest distance between two points at which the points still can be separated, distinguished as separate entities. So it is depend upon the quality of lens and the wavelength of illuminating light. The shorter the wavelength, greater the resolution. And next is contrast, which reflects the number of visible shades in the specimen and is needed to make objects stand out from the background. And this, the highlighting between objects and background is achieved. We can achieve this by using various staining techniques. So as I previously described that there are two type of basic microscopy. First is light microscopy and second is electron microscopy. There are further classification is there. Light microscopy, the compound microscope, bright field microscope, dark field, phase contrast and fluorescence microscopy are included. And in case of electron microscopy, transmission electron microscopy and scanning electron microscopy are there. The most familiar type of microscope which we are generally used in our labs is the optical or light microscope, in which glass lenses are used to form the object image. And light microscopy is a general term which we are used for any type of microscopy where light is being transmitted from a source which is on the opposite side of the sample to the objective lens. Generally, the light is passed through a condenser to focus it on the sample to have a maximum brightness. After the light has passed through the sample, it goes through the objective lens to magnify the image which we are viewed. A benefit of light microscopy is that it can often be performed on living cells, which cannot be performed by using electron microscopy, because we have performed electron microscopy under vacuum conditions. So the practical on living cells, it cannot be possible by using electron microscope, while in case of light microscope we can see a living cells also, like we can able to see division and migration of various uh, prokaryotes, bacteria under the light and compound microscope. So this is the simple pathway of uh, light in compound microscope, and this is the sample of compound microscope. So in light microscope, the light is visible light or ultraviolet rays, which we are used to illuminate our specimen. And two types of lenses are used in compound microscope. First is ocular lens, and second is objective lens. Objective lens um, um, could be a 4x magnifying power, 10x, 20x, 40x, and 100x. And ocular lens could be 10x or 5x magnifying has a magnifying power. And resolution and contrast are controlled by condenser and iris diaphragm, which are provided in uh, microscope. There are various applications where we can use compound microscopes and the other microscope. Here I have only given the applications of compound microscopes. So compound microscopes is used for viewing very small specimens and used to visualize different samples at high magnification power. The range of magnification power in compound microscope is 42,000 times. Uh, Compound microscope is used to observe both prokaryotic and eukaryotic cells. We can also uh, observe division of cell stages and migration of different bacterial cells. We can also use this for study different type of cells and cell structures, and it is uh, playing a very important role in the case of clinical laboratory to detect microorganism directly in the specimens and to characterize the growth of microorganism in different types of culture media. So this is all for today. Thank you so much.